Have you ever wondered what kind of products are used to harvest the food you eat? What happens if these products bring risks to people and the environment? Can we do something about it?
In my place of origin, Cosalá, Sinaloa, corn farmers face a significant challenge from the fall armyworm, which can lead to harvest losses of 10 to 100%. To combat this pest, farmers often use pesticides that cause serious environmental and health issues, negatively affecting the consumption that help regulate climate. In Mexico, pesticides are the second leading cause of poison deaths. For example, my father experienced health problems from using conventional pesticides, illustrating the tough choices farmers make between protecting their crops and their health. This motivated us to develop a safer solution to this problem.
While we were investigating, we noticed a significant increase in the price of corn. Recognizing that global factors can affect corn costs, we reached out to local producers to understand their situation. That's when we learned about the threat of the fall armyworm and the deaths and illnesses related to the pesticides used to fight it. After doing field research with diverse stakeholders, we chose to focus on creating a biopesticide that targets the fall armyworm, an eco-friendly alternative that would not only support sustainable agriculture, but also protect the well-being of farmers and consumers. A biopesticide was chosen as a project since it will be able to have social acceptance, independent and viable application on the field, responding to the producer's needs and the possibilities for different levels of production, and it will be able to have a positive impact without bringing conflict with political, ethical, or cultural interests. Therefore, our aim is to make a difference in the corn producing regions of Mexico and ultimately to provide a safer, more sustainable future for our agricultural communities.
Protecting maize is more than just protecting food, it's about preserving the heart of Mexican identity. Maize has shaped Mesoamerican culture for millennia, from tricycle to the sacred carving of corn on Aztec art. For Mexican, maize represents history, tradition, and resilience. It's in our daily tortillas and in our ancient history. By safeguarding maize, we honor not just a crop, but a living heritage.
We generated the ecological niche model for the fall worm using machine learning with the maximum entropy strategy. We projected this model to different future scenarios and concluded that in the case of drastic climate change, the habitat suitability of the worm will increase in some of the countries with the biggest corn production today, like the USA, Argentina, China, and the EU. This highlighted the importance of facing this problem and its growing consequences.
Our project focuses on creating a biopesticide using piperamides derived from black pepper. Piperine and piperamide has shown promising effects against fall armyworm larvae, but we aim to produce a safer, more effective alternative. By studying the biosynthesis pathway, we found a new piperamide with lower skin permeability and easier synthesis. To achieve this, we identified candidate enzymes for key biosynthetic steps through differential expression analysis. After obtaining inconclusive experimental results, we expanded and redefined our selection, validating potential enzymes through molecular docking and molecular synthesis simulations.
In order to elucidate the metabolic pathway that produces piperamides in the black pepper, a genome annotation was performed to get a function of the genes present. With this, we could reconstruct the metabolic pathways present in the plant, and specifically the phenylpropanoid biosynthesis pathway that is related to the synthesis of piperamides. With RNA-seq data, we performed differential expression analysis among different plant tissues. We selected candidate enzymes for the designed piperamide biosynthetic pathway by choosing genes highly overexpressed in the mature fruit and with an annotation corresponding to expected reaction. With the selected enzymes, we perform molecular docking as an additional evidence for the expected activity. In this way, we could assess if the proteins would bind to their ligands in a strong way.
With the aim of getting a bigger pool of candidate enzymes, an homology search was performed in different public databases using as query the sequence of previously selected enzymes for each step of the design pathway. This pool of alternative enzyme candidates was clustered in groups according to their similarity. It was from the most representative sequences of the clusters that the enzymes with the highest similarity to those originally selected that we took a subset of alternative enzymes for the next step.
To optimize our metabolic pathways for piperamide synthesis, we began by identifying enzymes that might potentially catalyze the target reactions. Out of over 120 candidate enzymes, we used molecular docking to predict binding affinity between the enzymes and substrates. This approach provides insights into the possible binding affinities and confirms the importance of the cofactor in some enzymes. However, since biological molecules are dynamic, we employed molecular dynamic simulation to further analyze these interactions, focusing on 38 enzymes from group 5. This simulation offered a realistic model of enzyme ligand complexes over time. Key parameters such as RMST, RMSF, hydrogen bonds, and total energy were analyzed to ensure stability. The results revealed a very stable interaction and energetically favorable complexes of 6 of these enzymes. This computational approach informs our next step, the experimental validation of selected enzymes to enhance piperamide synthesis in our metabolic pathway.
The expression of the genes will be controlled by a constitutive and inducible promoter, PIGAL, creating a temporal expression program for optimal protein production. These genes will be expressed in a polycistronic-like manner by using A2 peptides that allow the translation of different CDS from the same mRNA. The constructs are designed to be expressed in Saccharomyces cerevisiae.
We model reactions using differential equations. We simplify the process focusing on 3 enzymes that convert ferulic acid and piperidine into piperamide. This helps us understand deeply the equilibrium behavior. We compare 2 models, a single-strain system, multi-strain system, and a cell-free system. The SSF proved most efficient minimizing metabolite losses, while MSS is easier to manage short-term. Our goal, optimize production using an SSS.
To ensure that our engineering strength can effectively produce piperamides, we conducted cytotoxicity assays to determine the impact of piperamides and ferulic acid on cell viability, using both automated cell country and flow cytometry. We assessed the effects of different concentrations of these compounds. Our results revealed that piperamides exhibit significant cytotoxicity at concentration above 0.2%, with a sharp decline in cell viability at 0.6% and higher. Ferulic acid in contrast showed a mild impact on yeast cells, maintaining around 84% viability at tested concentrations. These findings guide us in optimizing production conditions, ensuring that piperamide synthesis in yeast remains efficient without compromising cell health.
We performed phytotoxicity tests of piperamides on maize, seed germination, and seedling development to determine their suitability as biopesticides in maize cultivation. We treated the soil with different piperamide concentrations and compared the number of germinated seeds to the control. The germination assays indicate that the control group has significantly higher germination rates compared to all piperamide-treated groups. That's why alternative strategies must be considered to mitigate these effects, such as formulation adjustments, application timing, combination with sweeteners, or alternative agricultural techniques.
To validate the real-world effectiveness of our biopesticide, we conducted a field test in Cosalá, Sinaloa, Mexico. This trial targeted the fall armyworm. Piperamides extracted directly from black pepper were applied to plots of the local San Juan corn variety. The test area spanned 300 square meters and was divided into 3 plots to compare treated and untreated areas. The results demonstrated a significant reduction of the fall armyworm population in plots treated with piperamides, showing about 50% fewer larvae compared to controls after 3 days. However, the effectiveness diminished after 6 days, likely due to environmental factors like rain and compound degradation. These findings provide valuable insights for optimizing the concentration and application methods of our biopesticide to enhance its persistence and efficacy in real crop environments. We used several computational and bioinformatic methods to design the project from different approaches that worked in synergy to be able to develop a good engineering cycle that allowed us to reach a final design suitable for experimental tests. To understand the overall situation and come up with a proper approach, we addressed diverse stakeholders. In our interviews with farmers, we uncovered key insights that guided our project. Participants, all male with an average age of 53 years, came from farming families and entered the field at a young age. Farmers identified the fall armyworm as a frequent and challenging pest in their cornfields, along with whitefly, wireworms, and stem borers. Although they recognize the risk of pesticides, they feel they have no other options. Alarmingly, only half of them use proper safety measures when applying these chemicals. Many resort to overuse or even mixing of pesticides, increasing environmental and health risks. When introduced to the idea of biopesticides, 90% were open to trying them as long as they are safe and effective. This feedback is crucial in designing a product that truly meets their needs.
We collaborate with Symbio 100 where Richard and Sakti provide us with linear extraction module, and we made it accessible to 170 high school students from Villalgo, Jalisco. With agriculture engineer Samuel Hebb, we learned that applying the biopesticide during stages B1 before is essential, targeting at the top of the plant where the fall armyworm attacks. AgroPeak can be part of our integrated pest management strategy, complementing practices like soil disinfection and chemical treatments for pests. Finally, we figured that we could also implement this pest management into a broader strategy seeking for shift to sustainable cropping systems.
Agroecological transition strategies and sustainable cropping systems gradually replacing conventional ones have been studied by local governments along with private enterprises like Dirección de Desarrollo Rural in Guanajuato and developed by social organizations such as PETATE, Plan Territorial de Transición Agroecológica de Tlaxcala, who helped us a lot in understanding both the problem and possible approaches for an integrated solution. The overall situation, the magnitude of the problem, and our proposal were taken to the Senate of our country, addressing our political leaders with the help of México Colectivo. To summarize, we came up with two main approaches, a direct and a deep one. The direct impact is given when the farmers and the environment stop being affected by conventional pesticides. The deep one comes when we use AgroPIP along with an integrated pest management strategy for a cropping system transition plan, where a biopesticide would help farmers shift from crops dependent on agrochemicals to sustainable ones, where worms won't be a pest.
The process, the instruments, and tools used, as well as the ethical and practical background, are explained through the project. The situation is deeply analyzed, and the research goes through different aspects and stakeholders. The solution proposal addresses the consideration of the different backgrounds. Such proposals are built upon the perspective of diverse stakeholders, from farmers and organizations to government and academics.
Our commitment to sustainability extends beyond biotechnology, aligning with the United Nations Sustainable Development Goals. This is reflected in our winning of the Climathon 2024 at UNAM, where experimentalship and founding help us integrate these principles into our project. AgroPIP protects corn crops from fall armyworm, reducing losses and ensuring a steady food supply for vulnerable rural communities while enhancing sustainable productivity. As a biopesticide from natural compounds, we support it by eliminating harmful chemical pesticides, safeguarding farmers and nearby communities from serious health risks. Preserving maize implies passing down indigenous knowledge, blending tradition with science for sustainable agricultural education. Safeguarding maize preserves cultural heritage, fostering community pride and inclusive communities.
Aligned with sustainable consumption and production, we minimize harmful chemical releases and promote the sustainable management of agricultural resources, benefiting soil, water, and air quality.
We actively promote climate action by advocating for agricultural policies that combat climate change and enhance awareness of sustainability practices. Protecting the biosphere ensures ecosystems that are resilient to climate change.
Finally, for Life Online, as a center of origin of maize, we Mexicans have the responsibility to promote biodiversity by providing safe alternatives to pesticides, protecting agricultural habitats, and conserving ecosystems.
Despite all the potential benefits of AgroPIP, it is still, after all, a pesticide. That's why we're committed to solutions that ensure its safe and effective use. We're developing precise application methods thanks to our field research and future implementation plans. We're also integrating AgroPIP into an integrated pest management approach, considering pesticides as a last resort option when there are already well-known mitigation strategies such as crop rotation, biological control, and legal action against harmful pesticides. Our goal is a sustainable solution that protects both people and the environment.
Through consultations with key stakeholders, including Climathon advisors and relevant political figures, we gather valuable feedback that shape our development process, ensuring our solution addresses real agricultural needs. Using an ecological niche model, we identified the potential future impact of this pest if climate change persists. AgroPIP offers not only a safer alternative to conventional pesticides, but also promotes sustainable farming practices, enhancing the health and livelihoods of farming communities. AgroPIP is a project with significant growth potential, aimed at making a meaningful impact on farmers' lives. To achieve this, we have developed the next strategy. Short term, we want to conduct laboratory tests on candidate enzymes provided by ginkgo bioworks. Medium term, broaden the scope of interviews to gather more comprehensive data and perform subsequent analysis. Conduct additional field tests with bean and corn crops using AgroPIP to verify improvements in results. Long-term, optimize a strain by integrating the complete biosynthetic pathway, ensure continuity of the comprehensive pest management project, and develop a transition plan for adopting a sustainable crop model.